Hello, a very good morning and good afternoon to you all, depending on where you are joining us today. Welcome to today's seminar. My name is Joy Kao. I'm the Regional Marketing Manager for Asia Pacific from LGC Biosearch Technologies. Before we jump into introducing the speakers and the topic for today, let's just go over some of the housekeeping rules. So all participants will be muted to prevent background noise during the presentation. We do have a few polling questions designed, so do encourage you all to participate in those questions as we are keen to understand your needs and research projects. There will be a dedicated Q&A session after all three speakers have finished their presentation. But as always, you can enter your question in the question window at any time during the webinar. We try to answer as many questions as possible during this Q&A session, but do rest assured, any questions that we don't have time for today will be addressed via either email or other channel. And of course, after the webinar today, we will also send an email with a link to the recording. Once again, thanks for joining us today. And let's jump right into the webinars. Today, we're really very privileged to have three SuperSHEroes, women scientists joining us today to share with us how they're innovating genomic technologies to accelerate their local breeding programs and to seed global change. So they are Dr. Sugana Aparo, Manager of Molecular Breeding Laboratory from Saindabi Plantation Technology Center, Dr. Inga Gazandem, Researcher from Plant Breeding Agricultural Research Council, Vegetable and Ornamental Plants, as well as Dr. Santatra Velumanantwa, Crop Improvement and Plant Health Scientist from National Center of Applied Research for Rural Development for FIFA. So, and of course, I'm your moderator for today. So let's first travel to Malaysia and hear from Dr. Sugana on how Saindabi Plantation is improving sustainability and knowledge within the oil palm industry by increasing genotyping efficiency with an end-to-end workflow. Dr. Sugana Aparo joined Saindabi Plantation, SDP, as Senior Scientist in the field of molecular breeding in 2011, right after obtaining a PhD in plant genetics from University Kambasa, Malaysia, UKM. She was responsible to set up a high-throughput genotyping facility at Saindabi Plantation Technology Center, SDPTC, Sedang, Malaysia in 2015. The setup positions SDPTC among the top echelon of the world's genotyping facilities and made possible the Edison Award 2017 New York under Energy and Sustainability category for SDP's Genome Select Innovation. Her significant achievement in the Genome Select project brought her the SDP Employee Award in 2016, followed by Best Publication 2018 award. Currently, she manages the Molecular Breeding Laboratory at SDPTC, which has recently received MSIEC/ISO 17025-2017 accreditation for nucleic acid testing to provide genomic solutions for oil palm planters. Dr. Sugana, please, the stage is all yours.
All right, thank you, Joy, for the kind introduction. Good afternoon, everyone from Malaysia. My name is Suguna, and I guess it has been a good 6 years being partnership with Biosearch right now for Saindabi Plantation, and really thankful to Biosearch for this opportunity to be able to share what we do at Saindabi Plantation. So today I would like to share on our one of the projects that's very close to our heart. It's called Genome Select. The idea behind this project is to increase oil yield for palm oil, as well as to implement sustainability and increase efficiency for palm oil. So in terms of palm oil, it comes from oil palm, which is a plant. And why oil palm? We know that many of us are already well aware of about the food gap that's coming soon in 2050, in the next 2 years, and there is a great demand to double up the vegetable oils in the next 30 years. So how oil palm can help to address this issue. Perhaps its potential to close the food gap, knowing that globally the oil palm harvested area is actually one of the least compared to other oil crops. Okay, so if you look at this slides with this graph here, despite being lowest in terms of harvested area, it has 10 times more of oil yield potential compared to even soybean, let's say. So not just that, on top of it, oil palm has 10 to 12 metric tons in each hectareage of potential in terms of oil yield. So obviously, we can see that oil palm is very efficient and productive in terms of an oilseed crop. So why not oil palm, right? So at Samdabi Plantation, our aim is to be a leading sustainable palm oil company, and so to date we are the world's largest producer for certified sustainable palm oil, or we call it CSPO, and we do supply essential ingredients to most of the famous brands out there. You are very familiar with these brands, I'm sure, Nestle, McDonald's, and so on. So we are present in 4 producing countries, mainly in Malaysia, Indonesia, Papua New Guinea, and Solomon Islands. And apart from oil palm, we are also having some businesses in rubber, cattle, and sugarcane. Okay, so Samdabi Plantation has this stand against deforestation, and we are very clear on this, especially, and we have all policies implemented, and a lot of engagement is currently going on with NGOs and also stakeholders, including planters, smallholder planters, how we can help to address deforestation issue. So in terms of how we can help, so that's where the Genome Select project came into the picture. So in 2016, we launched this Genome Select project. So what it does is we use genomic selection as a tool to analyze the oil palm DNA, and by using this tool, we are able to select for high yielding planting materials. So why is this important? So when we talk about deforestation, it's all about opening new land banks, right? So how can we address those issues? How about having a land that without having to expand the current land bank, but we will be able to increase the yield? So the only possible way would be, one of it would be coming up with new planting materials. So in terms of coming up with new planting materials, that's where the key is, in terms of coming up with a higher yielding materials. So that's where the technology comes in. And so in our plantation of Samdabi, in terms of coming up with best planting materials, we have come up with this idea of Genome Select. We have screened all the planting materials, and we have planted them on the ground, as I can show you next. These are the steps that we have taken in place. So oil palm genome, it has about 1.8 gigabase of size. The genome itself, it was sequenced in 2009, and what we did with the genome sequence, we have actually studied about the genotyping array and understand the backgrounds behind it, and we also tried to come up with the oil palm SNP chip specifically for oil palm, using Illumina arrays, and this work has been done and published in 2016. And then 2017, we also did some work on 2016 we did some work on the genome-wide association study as well, to understand the backgrounds and the genetic potential of our materials. And then along the way, this exercise, what we noticed is our populations has vast differences. And what we could do with this next is understand where this differences coming, and then try to see the causal genes, whether they could help us to address the high yielding material needs or not. And then we also noticed for oil palm, the LD decay rates is pretty fast compared to rice and millet and so on. So then we moved on to understand, okay, perhaps the next strategy that we want to put in place is to predict accuracy of genetic markers, right? So using selected markers, we want to see whether we can maximize this. When we were doing this exercise, this is when we realized that, hey, actually, perhaps maybe we should just take the whole genome effect to understand what's happening in terms of oil yield. We don't really need to know where exactly these causal genes are, but perhaps if we take the whole genome effect, then we'll be able to identify those individuals that can be raising for high yielding, right? So this is what we call as the Genome Select project. So with that, we came up with the genomic selection model, and we were able to do this only thankful to our historical data kept by our breeders. We have about 10,000 palms in the breeding trials or with a full data set for oil yield data, and we use this information to come up with the training and validation populations. And then we apply the model onto these materials, and our model can give an accuracy of 65% of accuracy. And what this means is we could use this model to increase about 16% of oil yield for our planting materials. So 16% is quite a lot. 16% would mean extra oil enough to feed 15 million people. So that's a significant number actually. And we also try to identify what are the potential markers that we want to use for this genomic selection, right? So we identified top 1,000 markers related to oil yield traits such as oil per bunch, oil per mesocarp, and so on. Now, this work has also been published in 2017. For the genomic selection, we have used the RR blob. So if anyone wants to be interested with this study, can follow up in this paper. And so the work completed in 2016, and we managed to plant in about 100 hectare range. What we planted is the high yielding materials. We selected based on ranking. The top high yielding ones, we put them on the ground. So in 2016, we planted about 100 hectare range, followed by annually since then, we've been planting 1,000 hectare range. So this we intend by 2023, we want to completely plant our land banks with our genome select high yielding materials. So hopefully that could be done. But also along this experience, right, we realized that our planting materials that we put on the ground in 2016 found to be bearing fruits much earlier. So usually we expect about 1.5 to 2 years to start seeing these bunches, the ones in the middle black color. These are the bunches that comes up and the oil comes from the fruits. Fruitlets on the bunch. So interestingly, for these genome select materials, we start seeing fruits as early as 1 year. And so that was really exciting times for us. Like we were thinking, like, could we indirectly select it for early fruiting as well? So it did excite us at that point of time. So then just to share, how did we get there? How were we able to deliver this 100 hectare range, right, of genome select planting? So of course, we have to make the best crosses first. And then in order for this to happen, we actually applied about 80,000 seedlings. We sampled them, we brought to the lab, we barcoded them, and then we genotyped them with 1,000 markers for genomic selection, right? So that would mean 80 million data points actually being genotyped. And with that, we also design mobile devices and apps, and enable us to do the selection in the field easier. We just go and scan the barcodes in the field for the polybags, and it makes life much easier for the breeder, especially to make the selection. And this is a really a big effort to be working together with our R&D teams in terms of operation, logistics, IT. So a whole big company came together and got this project delivered on time. And the fruits of that labor we see in 2020 this year, where we were expecting 16% of increase, but today we saw about 20% oil yield increase in reality. So this is after 4 years of planting. So this is a very exciting news for the team actually, right? So I think the major part of the success, I would call it a success because I think the major part of it also credit to Biosearch for helping us to set up our lab for genotyping system, where we opt for CAST. It is for us is easy, and we felt that that's the best way to go. And it's also at that point of time it was cost effective, flexible, and we were liking to have something low throughput all the way to high throughput. So that fits our intention as well. And so in terms of principles of CAST genotyping system, it will require mixed assays as well as sample DNA to make the reactions happen. But our approach, STP's approach to make that happen in our lab was, we know we need 1,000 good markers in the lab, right? But so we designed 2,000 before that, and we checked for the amplifiability in the lab and consistency. Then we finalized the best 1,000 with the criteria, certain criteria, for example, 90% core rate and above, and so on. We also introduced negative controls and also positive controls to check for QC. And on top of it, we also done some modification in terms of the volume required for the CAST mix. So we also did some cost savings in the lab, and we tried to work things out, and it worked. That was possible. And also, having an outsourcing option is always good, because if when things didn't turn out well, we always have a backup to fall to. And our equipment also comes from LGC's Nipline platform. And so we also worked out a good service contracts with LGC, who's now Biosearch, of course. And that really helps this whole project to be completed successfully. Okay, so for us, the SNP genotyping using CAST assay did provide a high quality and reliable method for our oil palm genotyping. So this is where the setup is in our lab today. This is how it looks like. So from it's a translation from discovery to deployment. We were able to do 80 million genotype data points in 10 months to keep this project done. So the story behind this is we established our lab in January to May. It just within 5 months, we got our people trained and all the setup or equipments commissioned in the lab. And then the next thing, May onwards, we keep running. That's in 2015. And April 2016, we put the plants on the ground, planted. So work done, accomplished. So today, this lab is considered one of the highest throughput in Southeast Asia, and it's also ISO certified. And we also implement certain important methods to keep the quality improved continuously, and such as method validation, quality screening, and also inter-lab comparison testing with LGC, the creator lab. Okay, so as I mentioned earlier, along the way, we also have devised some mobile apps and also tools to check the palms on the ground, to go and scan the barcodes. And by just scanning the barcodes in the field, we were able to tell whether they are the selected ones, what is their oil yield prediction like, and whether they are legitimate pure parental samples or not, and what are the codes of their parental palms. So all that is easily now visible on the field using a mobile app rather than going through a long paper kind of records, right? So this is really a useful tool for us. And the genome select fields that we have today are all geotagged. So each coordinate of their the palms that we have in the field is known to us, and the location is well recorded. Okay, and I think the highlight, as mentioned by Joy, for us would be the recognition that we have received for this project. Edison Award in 2017 for Energy and Sustainability category, as well as Best Product Innovation by Dutch British Council for Sustainability category as well. So in 2017 definitely was an exciting time for the team to be able to see the fruits of our labor indeed. So what's now? Today, we are trying to ensure that all our fields are planted with high yielding these genome select materials. And hopefully by 2023, we're hoping that all our land banks are fully planted with these materials indeed. So just to wrap up, for us, we think that this genome select is the first commercial success in oil palm industry, and this can lead to a day where deforestation is no longer required, but yet we're still able to feed the global population growth. Right. So, as I mentioned, 6 years of working with LGC or BioSearch today, we have come a long way, and we are a very happy team, and we had a very good experience working with you guys. So, indeed, thank you again for this opportunity to be able to share our part of the story to you. And talking about deforestation, I think we still long way to go. There's a lot of efforts currently. The company is trying to work with different NGOs and other stakeholders, and we are very serious about getting this in the right shape moving forward. Yeah. So this is the team. They have done all the work. My bosses, Dr. Hari Krishna, Dr. David, the molecular team where I am, and our scientific consultants, oil palm breeders. Of course, without them, this wouldn't happen. Bioinformaticians, tissue culture, seed producers, IT group, as well as crop protection and agronomy. So we all came as a team to make this happen, actually. So with that, I think thank you for listening, and I really thank God for this good experience that I had in this company. It has been a very good 10 years journey in this company. So I'm glad I could share this with you. Thank you.
Well, thank you very much, Dr. Sugana, for the wonderful presentation. It's really exciting to hear you share, the fruits of the Genome Select, the high-yielding oil palm, and really glad to hear, the partnership between us has been really successful and a great experience as well. Let's now move on to South Africa to hear from Dr. Inga on how they're applying agrigenomics and new molecular breeding tools to fortify potato varieties in sub-Saharan Africa, strengthening the local communities and addressing malnutrition. So Dr. Inga has been a researcher at the Agricultural Research Council, ARC, Vegetable and Ornamental Plants, VOP, since 2001. During this time, she has applied a variety of molecular biology, genomics, viromics, virus diagnostics, and molecular marker tools to mandated vegetable and indigenous flower plants. She also has performed plant transformation to improve crops for drought and virus tolerance. Inga graduated from the University of Pretoria in 2000 with a Bachelor of Science honors degree in biochemistry. After joining the ARC, she completed a master and PhD in plant biotechnology in 2012 from the same university. In these two studies, she characterized transgenic potato for fungus tolerance and identified various cowpea genes responding to drought stress, respectively. Functional characterization was carried out by transforming Arabidopsis thaliana with a selected drought-induced cowpea gene. Her current interest is in incorporating molecular markers to modernize breeding of various vegetables at ARC VOP. She is an alumni of the UC Davis African Plant Breeding Academy Class 4 last year in December. She is involved in fingerprinting potato and sweet potato for genetic trueness to type and marker-assisted selection for male sterility in onion. So with that, I'll pass on to Dr. Inga for her to share with us her exciting work. Dr. Inga, please.
Good morning, ladies and gentlemen. Thank you, Sugana, for this very impressive presentation that you gave. I would like to come in with the part that I'm still new at to use the services by LGC BioSearch technologies in the migration of our potato fingerprinting system from what we're currently using, microsatellite markers, to a panel of single nucleotide polymorphisms. So the contents of this presentation is to give you briefly a background on the crop potato, the rationale of why we wanted to change this approach, and then how do we plan to use LGC BioSearch technologies in doing this change, the anticipated outcomes, and then shortly the benefit that we expect for our country. So as a background, the potato crop, for those of you that are not knowledgeable, it was domesticated more than 8,000 years ago in the South American continent. And there's more than 100 and up to 180 wild species known. It was introduced to the rest of the world in the 1600s and is now grown in more than 150 countries around the world. There are at least 4,000 varieties of potato known worldwide, and they are categorized in the different categories of processing or fresh consumption use. And it's also one of the 4 major staple crops together with corn, maize, and wheat. In South Africa, it's the largest vegetable commodity, so it's quite important to our farming industry. So the background of the genetics, it is vegetatively propagated, and it's highly outcrossing and therefore very heterozygous. So pure line breeding is not possible for this crop, because if you try and inbreed them, they lose their vigor and they stop flowering. So the improvement of potato is through the hybridization of parents with good traits into F1 families. And then after the progeny that have the beneficial traits are selected, they are clonally multiplied. But you require multiple years of multiplication before you can establish replicated field trials. So currently DNA fingerprinting, as a background, what this technique is for, is to identify the genetic differences between cultivars or lines, so to determine the clonal identity, and for genetic purity testing or tuners to type. And this is important in situations where cultivar protection, which is becoming more and more important. So the old method that is currently being performed at our institute is a PCR-based method of amplifying simple sequence repeat sequences, which then generates a mixture of fragments that are then separated on a gel matrix according to their size. So the result is a characteristic pattern or fingerprint, which is then compared to a known reference. So it's different sizes and also the absence of presence of different SSR alleles that you look at. So this is an example of a very nice executed page gel of the fingerprints from one marker, and it shows the differences between here example of 5 cultivars. So South African potato industry is very dependent on our fingerprinting services, because the trueness to type information is important when the seed producing farmers produce seeds for farmers, and also when our in-house in vitro multiplication facility multiply lines, they need to be sure of the true to typeness of the clones. And our laboratory is the only one currently in South Africa that provides the service. But as a rationale for this change in project is because PAGE is such a low throughput and labor intensive method, and quantifying the fragments is very subjective. We thought it is an outdated approach and it needs to be migrated to a new technology by phasing out this old technology. And SNP panels, or single nucleotide polymorphism panels, are suggested by many authors as a probable approach due to the ease of genotyping and also cost and throughput considerations. So just shortly to summarize some disadvantages that we experience on a daily basis of doing SSRs for fingerprinting. It's a very low throughput technique and high cost of chemicals. And we have the problematic scoring problem where, because potato is a tetraploid, you have multiple alleles, but also stutter bands are caused by the polymerase that you can't do anything about. So you have a very subjective sizing situation, and the calling of alleles and their exact sizes is a problem. So it's difficult to generate an accurate allele database. In comparison to this, the advantages of SNPs is because it's the most modern method for detecting genetic polymorphisms, you can do it at a much higher throughput and lower cost, but you also see that it's very flexible in how high throughput you want to go. And then the SNP calling is very robust and accurate and much less subjective. It is more robust that it can work from more crude genomic DNA extractions, because specific primers and probes cause the reaction to be very specific. And then it is also easier to assign the putative cultivar identity because your database is simplified by a more binary system of a SNP genotype. So the proposed approach, where we've decided what protocols we have to our possible to choose from, there is a SNP array for potato. The commercially available one has a bit more than 8,000 SNP positions, but there's also a 12,000 one that's not commercially available. But the place that we could have a genotype that had a much higher cost, that we couldn't consider that. So we chose between the two technologies from LGC Biosearch, SeqSNP, which is a targeted genotyping by sequencing technology, or also CASP assays. And in our investigation, the initial method development, we could genotype many more SNP positions for the same price using SeqSNP than doing CASP. But then the process following, we will definitely be using the CASP assay, where we will do the in-house SNP genotyping and not be dependent on sending samples for genotyping in a different lab. So as the method development, the first applied way of SeqSNP is first we had to choose a diverse list of commercially important cultivars and South African breeding lines. The process then was to select using SeqSNP to genotype them in all these diverse lists, and we used the all-inclusive service at LGC Biosearch Technologies. So my samples are not yet sent, but all the preliminary work of choosing the cultivars and the SNPs has already been done. So the SNPs was evenly spaced over the 12 potato chromosomes, as this graph indicates. So by LGC Biosearch Technology, I expect that the SNP calling will score them into the 5 gene dosages, since potato is a tetraploid. And then from that, we'll choose the informative SNPs with the polymorphism information content to be as high as possible, where we then choose the optimum SNP panel. And this is where the SeqSNP is an important approach, because it gives us the sequencing information around the SNP of interest. So we can choose SNPs that don't have any neighboring SNPs, and also avoid SNPs that have flanking region that is 280 rich or GC rich, because the next step would be to validate this with COSP assays. So the COSP assays will be designed using this SeqSNP sequence information of the selected SNP panel, and that will validate the sequences. And if the COSP assay is effective, it's not expected that all of them will be 100% functional. And then we can in-house determine the endpoint fluorescent assays using a real-time PCR machine. So this will enable us to set up the SNP genotype database for our whole list of South African potato cultivars. So then when the method is applied, when we get samples sent in, we can then apply the COSP assays of the sample submitted for fingerprinting or purity requests, simply compare them to the database, and then assign the putative cultivar identity from our database. So the anticipated outcome using LGC Biosearch technology is, as I've said, the SeqSNP provide the flanking sequence information applicable to our compilation of genotypes. So we are not dependent on only the sequence genome of potato, but we can see exactly what is going on in our collection. And then the electronic database of SNP genotypes, I feel, is a very important resource that we can be using going forward for identifying cultivars. So in-house, the COSP assay of the submitted samples, we expect it to be more robust and accurate than the current system, and also less subjective because there's no guessing of the fragment size. So the SNP panel will be a useful resource to resolve identity or mixing issues by you can identity call the unknown sample as long as it satisfied the predetermined similarity cutoff threshold. So the benefit to our country is a streamlined potato fingerprinting service using a more modern technology, and it will increase our efficiency, reliability, and sensitivity with a much higher throughput than currently, where we can only analyze approximately 30 samples per day with the page process, which is actually a multi-day setup, while with the COSP assays, you can do multiples of 96 samples and read them on a real-time PCR machine. So if the development costs have been completed, we don't expect it to have any drastic price change, and it might even be fewer if only a few SNPs need to be selected from the full panel if you're only required to distinguish between 2 known cultivars. With this, I would like to acknowledge the company that I work for, Agricultural Research Council, and then also the funding body, Potato South Africa. Thank you for your attention.
Great. Thank you, Dr. Inga, for sharing with us how your plan and your work on setting up the potato fingerprinting service locally. And let's now, last but not least, travel to Madagascar to hear from Dr. Santatra on how they are improving the biofortification of the superfood amaranth through genomic selection to maximize the nutritional intake for the rural communities. Dr. Santatra is Malagasy woman scientist, interested in various life science areas. She joined the Department of Agricultural Research of FIFA in 2017. Santatra manages research programs and carries out research focusing on selection for disease and pest resistance, plant pathology, and biological control. She also leads the plant health laboratory and takes part in the design and the development of large research projects through national and international collaborative research programs focusing on food security and fight against malnutrition and pest and disease epidemic surveillance and biocontrol in the Southwest Indian Ocean. Santatra has over 13 years of experience in areas of agriculture-related R&D. Previously, Santatra worked for a decade at the National Research Center of Manna. She was the head of Wheat and Triticale Research Unit and responsible for the selection of the superior wheat and triticale cultivars. Santatra is also a graduate of the African Plant Breeding Academy Class 4. With that, I'll pass over to Dr. Santatra. Dr. Santatra, please.
Thank you, Joy. Hello, everyone, and thank you, LGC, for inviting me for this webinar. It's a great pleasure to share you today how I am going to use the genomic selection to assist the parental selection of amaranth towards the development of biofortified cultivars. To begin with, why breed amaranth? Malagasy population is food insecure and chronic malnutrition is widespread at the national level with an adequate daily intake of nutrients such as protein, minerals, and vitamin A. Nutrient supplementation of vulnerable groups needs to be strengthened. In light of this situation, amaranth can provide affordable and significant amount of proteins, vitamins, and minerals that are often missing from the diet of vulnerable Malagasy population. Often considered as wheat, amaranth is valued as vegetables in Madagascar. Young and soft leaves are collected for consumption. Cultivation and consumption of amaranth are currently promoted to address malnutrition issues. Therefore, the breeding target is to develop amaranth cultivars enriched simultaneously with multiple macronutrients, targeting levels required for better nutrition. Thus, the product target is to develop cultivars that offer outstanding leaf yield and high level of iron, zinc, calcium, vitamin A, and protein. Also, cultivars suitable under several agroclimatic conditions and cropping systems, and cultivar for rural and urban target market customers. Let's take a brief look at some quick facts of amaranth. The genus Amaranthus is over 70 species distributed worldwide, and germplasms are maintained in approximately 61 worldwide gene banks. The amaranth species are highly self-pollinated. Seeds are mostly the means of propagation. Life cycle takes approximately 6 months. Amaranth species have a small genome size, about 500 megabase, and some genomic resources have been developed to assess the genetic studies. The reference genomes of 3 species are currently available, and nutrient content can vary across genotypes and according to the environment and growth stage. From a simple visual observation, Malagasy amaranth accessions highlight a morphological diversity. As the breeding programs for amaranth are just starting to be developed in Madagascar, the first stage should be devoted to pre-breeding activities for the purpose of broadening the genetic base. For an efficient breeding program, the judicious choice of source materials to be used in crossing to produce superior recombinant genotypes with desirable traits is a prerequisite. For the parental selection, we choose to use genomic selection approach using single nucleotide polymorphism markers or SNPs to do an accurate job or improving the desirable traits. Why go for genomic assisted parental selection? Traditionally, selection in plant breeding has relied on scoring phenotypic traits being evaluated in several environments, but the efficiency of phenotypic selection is reduced by genotype by environment interaction that can mask the expression of certain traits. Also, phenotypic selection is less effective for traits with low heritability. Those limitations can hamper the increase in genetic gain in the breeding program. So the use of molecular markers can maximize genetic gain and optimize selection accuracy. Marker-assisted selection, or MAS, which uses molecular markers linked to desirable traits to select a genotype, outperforms phenotypic selection. MAS have the ability to select for low heritability traits, suitable for traits controlled by a small number of major genes. MAS is, however, less effective when screening for more complex traits which are controlled by many QTL with minor effects. But many important traits are complex in nature and are controlled by a large number of small effect genes distributed across the entire genome. So to deal with this limitation of MAS, we will incorporate genome-wide markers to increase the chance of markers being in linkage disequilibrium with QTL, influencing the trait of interest, and to capture small effect loci. Genomic selection assumes that all genome segments contribute to the genetic variance and each segment is in high linkage disequilibrium with a minimum of one non-genetic marker. So, effect of all genetic markers across the whole genome will be estimated without having a precise knowledge of where specific genes are located and will be used for predicting genetic merit of selection candidates. In practice, genomic selection comprises three phases. First, the establishment of a reference population for which phenotypic and genotypic data are known and in which the marker effects will be estimated using a statistical prediction model built based on phenotypic and genotypic data. Second, the estimation of the breeding values of selection candidate individuals for which the genotypic data are available and fed into the statistical model to calculate the genome estimated breeding values, or GBV. And third, based on the GBV ranking, the selection of the most suitable individuals with target trait as parents for making crosses. In this way, genomic selection approach removes gene and association mapping. Now, how parental selection of amaranth is going to be done and how the use of genomics will be implemented to assist the parental selection. The year one of the parental selection step will be dedicated to germplasm assembly, acclimatization of exotic accessions, preliminary phenotyping and species characterization, seed multiplication for further step, and germplasm conservation. A large-scale sampling of local amaranth species will be carried out to capture broad genetic diversity and will be added to the current collection consisted of 18 national accessions. We expect to collect 200 new local accessions. Besides this, 300 accessions of elite inbred or advanced lines of Amaranthus cruentus that will include ideotype traits will be acquired from worldwide gene banks to broaden the genetic base. Acclimatization of exotic germplasm to local conditions and preliminary phenotyping will be carried out twice in rainy and dry seasons in open field. The morphological assessment will be based on 34 characters, which will be then used to analyze the diversity and the relationships among and between accessions. Then, assessor markers will be used to characterize phylogenetic relationships among amaranth accessions and to determine Amaranthus cruentus species among local accessions. Then, Here too will be devoted to phenotyping step. About 300 lines of Amaranth cruentus from the germplasm will be phenotyped in a screenhouse under conditions representative of major stresses found in the growing areas of Madagascar. Data for 18 quantitative traits related to foliage productivity and nutritional value will be recorded for 2 cycles in the rainy and dry seasons. Then, the data recorded will be subjected to statistical analysis to estimate the level of variation among lines, the genetic variances, and the relationship among traits. Thus, we have the phenotypic data. Here, 3 will be devoted to genotyping, data analysis, genomic prediction, and selection of suitable parent candidates. To obtain genotypic data, we will apply the genotyping by sequencing method, a reduced representation of sequencing methods for discovering SNPs in amaranth genomes. We will use a PK1 restriction enzyme to reduce genome complexity. And the raw GPS sequence data will be aligned with all available reference genome of Amaranth cruentus and analyzed with a computational pipeline to detect polymorphism in Amaranth accessions and to generate large numbers of SNPs for use in genetic analysis and genotyping. Molecular data will be then used to group all accessions. The genetic structure analysis will be assessed and to determine relatedness among accessions and the pattern of population structure of the reference panel of amaranth germplasms. Next, the genetic diversity will be estimated among lines and within each group. The value for the linkage between 2 different alleles will be estimated. Polymorphic SNPs with higher coverage will be retained, and we expect more than filtered 10,000 SNPs for the subsequent analysis. Then, genomic prediction for every trait will be performed to predict how useful each individual will be good as parent. For these 2 statistical models will be performed for prediction based on their assumption of the distribution and variance effects and according to the algorithms. First, we will use the genomic-based linear unbased prediction model, or GBLUP, which uses a genomic relationship matrix representing the genomic relationship between individuals. It is known that GBLUP assumes a common variance for all SNPs and is suited for traits influenced by a large number of minor genes. Second, we will use the Bayesian linear regression BASE-B model, which relies on marker effect estimation. BASE-B assumes that the majority of SNPs contribute no effect while a minority does. Thus, it is suited when trait is strongly influenced by fewer genes with large effects. The GBV of all genotyped individuals will be predicted by solving the mixed model equations. The accuracy of prediction models for all traits will be evaluated by cross-validation, and the predictive ability will be calculated as the correlation between phenotype and GBV, and the similarity of the 2 prediction models for each cross-validation will be estimated. Then, the model with the best predictive ability will be used for subsequent analysis. The GBVs will be then ranked and used to select the top genotype or lines of candidate parents, and we expected to select 20 and 50 top GBV genotypes. In short, genomic selection uses SNP genome-wide markers, including those with minor effects as predictors, allowing us to use GBVs to pick out top parents for the next breeding cycle, so as to achieve more reliable selection and to accelerate cultivar improvement of amaranth in the near future breeding stage. The next step will be cross-prediction using breeding simulation of the top candidate parents in order to identify all possible and suitable intercrossing plan to improve follower heat and nutritional content of amaranth simultaneously. To conclude, this proposed approach will be the first genomic drive selection for amaranth in Madagascar, and expected outcomes will be high-performing and nutrient-rich elite genotypes or lines, phenotypic, genotypic, and marker data, valuable knowledge of the local genetic resources, and prediction models that can be used directly to estimate the performance merit of new genotype germplasm without phenotyping. Overall, this first stage represents a good starting point to form effective strategies for the development of PO-fortified amaranth cultivars with an increased rate of genetic gains for valuable traits, namely follower yield and nutritional value. And the integration of such technologies into plant breeding programs can make a powerful step change in agricultural productivity for Madagascar. So, and this is how we plan to incorporate genomics into parental selection of amaranth. And I just want to say thank you for your time. Thank you.
Great, thank you, Dr. Santatra, for sharing with us your work, using genomic selection for parental selection. I'm personally very keen to understand more about superfood. I believe everyone is, during the COVID lockdown when you try to stay healthy and boost your immune systems. With that, let me then run into the final Q&A sessions. I believe we have one question for Dr. Sugana. So there's one question trying to understand, are there any trade-offs to the significant increase of oil yield observed in the genome select pumps?
Okay, thank you for the question. The only trade-off I can think of would be the cost involved in setting up the whole SNP line and the equipment, right, to do the genotyping. We invested quite a lot into it in the beginning, but I think it's still paid off because, as I mentioned, the increase came 16% more as predicted, but then in reality it was 20% that we observed. And even that 1% increase is actually significant, and I think we got covered our investment overall by doing this project. Perhaps the other thing that is as a form of challenge for us would be the sampling part. Of course, doing this large scale of sampling require a lot of manpower. Unfortunately, we don't have any tools that more sophisticated to do oil palm sampling. Because the leaves are very fibrous and it's difficult to punch them even using a handheld or automated system, which we tried a few options, but we were not able to get anything suitable to cover our sampling in a more robust or high throughput manner. So that is still, I would say, a challenge in the beginning. So we had to get all people come on board with us, like we are molecular breeding team, but we got our secretary, our finance, everybody came down and did the sampling along, in the field. So that whole experience was quite pretty amazing. So yeah, I would say the trade-off would be something to do with the sampling side of things and setting up the lab. But for us, I guess we got that covered along the way.
Thank you. Thanks a lot for sharing. And there's also another question for you, Dr. Sugana, since you've been talking. So there is a question trying to understand how much time was saved using a genomic selection approach.
How much time saved? Okay, so
Yes.
if I can go back in time, we did the modeling in 2014. It took us, say, about 3 months to get that sorted out. Our bioinformatics teams got that thing sorted out. And then 2015, we established our lab, right? So we established our lab with the equipment required. So we established in 5 months with training our people and so on. So then another 10 months to do the genotyping of 80,000 seedlings. So in less than 1 year. We got this whole process covered. But behind on the hindsight, there's the genome need to be sequenced. There's a lot of understanding of the genome. There's genome-wide association that we have carried out, so it can go all the way back to 2009. So we invested closely 7 years in just understanding the fundamental.
Great, thanks for sharing.
But sorry, I want to add that genomic selection seems to be the fastest way for us to get here. Otherwise, I think the other marker-assisted breeding tool seems to be taking longer time.
Great, great. I think we have time maybe for one last question. I guess is, the question is addressed to all three panelists. And I think Dr. Sugana has already shared some of the challenges that you are implementing genomic selection. So just keen to understand from Dr. Inga as well as Dr. Santatra, what's sort of the biggest hurdle that you face when you try to implement either genotyping workflow or genomic selection program locally. So can you share a bit with us, some of the challenges and how you might have, overcome those challenges with the partnership that you might be experiencing with us?
Okay, even if genomic selection or the use of molecular tool promises to be one of the next major advances in breeding programs, there are practical limitations for the implementation of genomics-assisted selection in Madagascar and in my institute. We would need sequencing or genotyping lab and more resources. And to overcome these issues, the collaboration and partnership with centers which have genomics facilities are important and helpful. For instance, we can use the genomic services and bioinformatic analysis services of GCs, biosearch technologies, to perform target genotyping by sequencing on a wide variety of amaranth, or also SNP discovery and SNP genotyping.
Great, thanks for sharing, Dr. Santatra. Dr. Inga, do you want to have a few words to comment some of your challenges that you're experiencing, how you might have overcome those?
Yes, sure. So the simplest challenge we have is funding, and the funding is because molecular genotyping, technically you need a lot of samples to be analyzed and the techniques may be quite expensive. So funding is our most basic constraint. But also to get to genomic selection, you need to have good phenotype data. So that also costs money. So we have all the bioinformaticians in our institute at the biotech platform, and we have access to service providers that can help with genotyping. So we don't need to necessarily do it ourselves. That's where LGC and other service providers come in. But in simple terms, I think resources in the form of funding for phenotyping as well as genotyping is our constraint.
Great, understood. Thanks a lot for all sharing, for all three panelists. I would once again, really give a heartfelt appreciation to all our speakers today for sharing with us some of their work they've been doing to accelerate local breeding programs, to see how, these three SuperSHEroes, women scientists, are really championing genomic and innovative genomic technologies to promote sustainability, efficiency, as well optimal breeding workflows locally. With that, I'd like to end, today's webinar. If we don't have time to address your questions today, be rest assured we're going to get them addressed via email. You can always reach out to us. I have my email address here. Feel free to reach out to us, and we'll be sending out a recording after this webinar today as well, so you'll get time to review for those. With that, I'd like to end this webinar for today, and enjoy the rest of the day, and thank you very much for joining us today.